Now we're live. So, what'd you write your paper about again?
About being a really nice girl.
Okay. How are we doing today, y'all? Good? Are we like wiped out from anything? Did y'all have anything today?
No, we didn't have anything.
No tests? Still lucky. Okay.
Yeah.
What chapter are y'all on now in this stuff?
Like a million.
Forever far?
We introduced a new G word today.
On 13? Awesome. Well, in theory, we're going to get through both chapters 12 and 13 today. So we'll be all caught up, and y'all can actually answer questions in class.
Yeah.
In theory. We'll see what happens. All right. How about, seems like energy's kind of low. How about we all just like get up, walk around a little bit, get ready. I'm serious. We're going to do it. Everyone stand up. Everyone stand up. We'll put some music on. We'll be like Dr. Russell.
Perhaps some Spice Girls.
Maybe not Spice Girls.
Spice Girls on your phone?
I don't have Spice Girls on my phone.
What do you got?
What do y'all want? What do y'all want? Someone give me a song. What's a hype song? Eye of the Tiger. We can do that.
We don't know how to get hype.
You don't know how to get hype? Y'all try one. Y'all should be hype off your freaking minds.
That's my pregame song.
All right, I need someone's phone. My internet's not working. Someone give me your phone. Type in Eye of the Tiger. Play it. I'm going to put it on the mic. We got to get excited.
Oh, wait.
They're telling 420 years.
We'll climb all the stairs like Rocky going up this way. We'll celebrate at the end.
Can I get 5 tokens? I'm looking this up.
Sure, you can do whatever you want. It don't matter.
He did a year in,
Yeah, I'll get
year in
Spice Girls on
Washington,
here.
year in Quantico or whatever. Yeah, he had to.
We all have to.
Okay,
There you go.
we took an audible. We're doing Spice Girls apparently.
He got his major. He's like major.
Spice Girls on
He's
jam.
like, gotta go to Washington for a year and, press flush, make the right plans. He made the right plans and came out and he was battalion XO.
What's happening in this video?
What you think about that? Now you know.
Everyone get up and dance. Even if you're white like me.
Are you for real? I won't be hasty. I'll give you a try. If you really bug me, then I'll.
All right, that's good. Someone embarrassed themselves, so that's good.
Shout out to you, Dr. Russell.
Yeah, I didn't want to steal his party in the USA, if that's okay. That's his thing. All right, so chapter 12, we are doing metabolism of other sugars and the pentose phosphate pathway, or PPP. So who can remember in their lecture what like the two biggest things are with the PPP thingamabobber? Anybody? Okay. We'll get into it. All right, so really what this chapter is about is saying, hey, there's other ways to get energy besides from glucose, right? Because we have other sugars in our body besides glucose. Maybe we got some fructose, galactose, mannose, what have you. So this chapter is all about, hey, let's utilize those things to make energy as well. So that's what we're going to be going through today. So some things to pay attention to as we're doing this. We're going to do the super zoom. All right, so first thing, we want to link the enzymes back to glycolysis to determine their function. So we're going to be seeing some other enzyme names, again, that we've already seen before, and that's going to give us a big key to what those enzymes are going to be doing. So really, we're just refreshing our memory as opposed to relearning new enzyme names. Well, as we're doing this, really need to know what the name of the first enzyme is of a pathway. and what it does. So just like we do in glycolysis, just like we do with the TCA cycle, and I guess to a lesser extent last week, got to know the enzymes, got to know what they make. So this chapter has about 5 different pathways in it, but what's really nice is they're a lot shorter than the other ones. So still some new things to learn, but not nearly as big and cumbersome as the glycolysis or TCA cycle. And lastly, sorry, there's a little bit of a glare. We need to know what the final product is that's being made. So just like at the end, we know kind of what's happening. So starting off, so other things that we use, like I said, fructose, galactose, mannose. Probably zoom out a little bit. And we're going to start off this chapter with fructose. So lots of our fruits have sucrose and fructose. We should know that by now that sucrose is glucose and fructose combined, right? Let's see, what's maltose? Anybody who remembers what maltose is? It's like lactose and glucose, maybe, or is that? Nope, never mind. All right, we can just skip that. Y'all gotta tell me. This is. I don't. In try 4, you'll get Dr. S's husband, Dr. Marsban, and you'll ask him a question, and he'll be like, I don't know. I guess I got to go study that. And really, what he's saying is, you need to go look that up yourself. So, anyway, all right. So here we've got sucrose. It's just saying that we can break it up into glucose, fructose. Really cute picture. And here is what we're going to be dealing with fructose. On the right-hand side of our page is everything in word form, but I like to start off with the picture form just because pictures are more fun for me. So first thing we've got is we're starting up here in the top left with fructose. So fructose can either go 2 places. It can either go into the muscle, it can go down this pathway, or it can go into the liver this way. So based on the enzyme, we'll determine the liver has a different enzyme that allows fructose to catalyze by the first reaction, and the muscle has its own enzyme. So in the muscle, we're using hexokinase. Right? We've already seen hexokinase. What does hexokinase do? Yeah, so in glycolysis, it takes glucose and makes it glucose 6-phosphate. But really, if we deconstruct what the enzyme's name is, hexokinase is a 6-carbon sugar, and we're adding a phosphate to it. So since fructose is just the ketone and is a 6-carbon ketone, whereas glucose was a 6-carbon aldehyde, right? So that it's really the same 6-carbon sugar. It's a very general name, hexokinase. So we can use that same enzyme to put a phosphate group on fructose, making fructose 6-phosphate. And that's really, and is fructose 6-phosphate a part of glycolysis at all? Yes, right? Because it goes glucose to glucose 6-phosphate to fructose 6-phosphate. So we've just made fructose 6-phosphate. So that just goes straight into glycolysis, gets broken down, follows the rest of the pathway. Done and done. So muscle, really simple. All it is hexokinase making fructose 6-kinase. Questions on that? So we need to know that muscle is hexokinase, and as we'll see over here, in the liver, it uses fructokinase. So fructokinase takes fructose and puts a phosphate group on the first carbon, making it fructose 1-phosphate. So there's kind of where it differs at. So it's in a different organ, or it's in a different organelle, I guess organ. So it's going to make something slightly different. So now we're at fructose 1-phosphate after putting the phosphate on with fructokinase. Then it's going to open up the chain. That really doesn't matter. But then we're going to go to fructose 1-phosphate aldolase. Who remembers what aldolase did in glycolysis? It was step number 4. That's pretty good. So it takes fructose 1,6-bisphosphate and creates the glyceraldehyde 3-phosphate and the dihydroxyacetone, right? So whenever we saw that, so that was just aldolase. So aldolase took something and broke it up into two. So that's exactly what this aldolase is going to do as well. It's going to take fructose 1-phosphate and it's going to break it up into glyceraldehyde and dihydroxyacetone phosphate. So really the same enzyme names, doing the same enzyme functions. So it's really easy to just apply what we learned earlier to what we learn now. Yeah? Yes. Cool. So now we've got, we got glyceraldehyde and we've got dihydroxyacetone phosphate. So this is, in glycolysis, we had glyceraldehyde 3-phosphate, right? And now we've only got glyceraldehyde. So all we need to do is just put a phosphate group on the third carbon, and we can just put this G3P right into glycolysis, right? So that's exactly what we're going to do. So we're going to have glyceraldehyde kinase. We're just going to add a phosphate to glyceraldehyde, making glyceraldehyde 3-phosphate, goes right into glycolysis. Really simple. So that was, I think, 3 enzymes. So we've got fructose. That by itself can't go into glycolysis. So let's try to see if we can rearrange glucose to get in there. So going into the liver, we're using fructokinase. Liver is fructokinase versus muscle hexokinase. Makes it fructose 1-phosphate. The 1 is a big deal. Then we're going to do fructose 1-phosphate aldolase. When we see an aldolase, we're thinking it's going to break apart into 2 3-carbon molecules, yeah? So that's going to make glyceraldehyde and dihydroxyacetone phosphate. Then with our glyceraldehyde, it's not quite glyceraldehyde 3-phosphate. So we're going to put a phosphate on it with glyceraldehyde kinase, making glyceraldehyde 3-phosphate into glycolysis. Really simple. So we've already got dihydroxyacetone phosphate. What enzyme takes that to glyceraldehyde 3-phosphate normally in glycolysis and here? Says it. Triose-phosphate isomerase, right? So that's step 5 in glycolysis too. So really simple, just reusing those enzymes just with fructose. So that's really all fructose is, breaking down simply in both the liver and in the muscle. So we can take it, so we could store glucose as glycogen, right? Well, we can also take fructose and store that as well. So we can take glyceraldehyde and put it through the enzyme alcohol dehydrogenase and make glycerol, which then can form lipids, which y'all will learn next try. But alcohol dehydrogenase will make glycerol, making lipids. But say we needed to break down glycerol, or we needed to break down fats for energy. So we can then take glycerol, add a phosphate to it, glycerol kinase, and that'll make glycerol 3-phosphate, which can then go over here and make dihydroxyacetone phosphate with glycerol phosphate dehydrogenase. So I'd say this half is more of the important half than this one, but it's just really cool to know how everything's connected like that. Questions on that? Is that making sense? Is that over anybody's head?
What does it say right there by number 2?
What did I say? I think I was going to try to prompt y'all with questions. What did aldolase do in glycolysis? So that was just a question I asked y'all. And so we can see here, what I wrote here too is if you have hexokinase, it puts the phosphate on the 6th carbon. So that does it with glucose, it does it with fructose, puts it on the 6th carbon. But whenever we got specific with fructokinase, it's more specific, so it's going to go on that first one, the 1st carbon. So the reason I point that out is because what he'll do on a test is he'll say, what does fructokinase produce? And he'll put fructose 1-phosphate, fructose 6-phosphate, glucose 6-phosphate, glucose 1-phosphate. So if you really don't know what each of them makes, specifically, then you're just going to be guessing, and it's not really fun to guess on his tests. We all experienced that with test 1. So, all right, so this is what we have in word form. In word form, do I need to go over this again? So in the muscle, we are using hexokinase. That's a test question. Making fructose 6-phosphate, I'm sorry. And the liver, we can't use hexokinase. So in the liver, we use fructokinase. Like I said, we can then take glyceraldehyde and make glycerol and store it. And then we can also take glycerol, make glycerol 3-phosphate, and then make that dihydroxyacetone phosphate glycolysis. So the whole purpose of this is just, hey, we need energy, but we have other things besides glucose. So let's get this oxidized so we can get that energy, take it through glycolysis. Because glycolysis, TCA cycle, oxidative phosphorylation gets us the most energy in our body. Right, really efficient overall. Cool. So that's fructose, all done with fructose. Next, we've got galactose. So galactose is a C4 epimer. And it's a milk sugar. I thought it was cool as a milk sugar, but he might ask you what kind of epimer galactose is, so that might be a test question. And it's a hydrolysis of lactose. Yeah, so lactose is galactose and glucose, I'm pretty sure. Bless you. And it requires 4 steps to enter glycolysis. That's just a nice little picture to say, hey, look, there's a C4 epimer, whatever, we're going to move on. So, picture form of how we metabolize galactose. So galactose is a sugar. We need to get energy from it. It's not glucose, it's not fructose. We need to find a different way to do it. Here's a different way to do it. So galactose, we're using galactokinase. It's really nice. We're just going to put a phosphate group on galactose. It's specific, right? It's not saying anything about how many carbons it has. It's galactokinase. So if it's specific, it's going to put the phosphate on the first carbon. So galactose 1-phosphate. Need to know it, need to know it, need to know it. So we've got galactose 1-phosphate, and this is where it kind of gets a little tricky. You've got this UDP glucose over here. So what's going to happen is we're going to have a transfer reaction, as we can see here by the enzyme transferase, right? It makes it really simple when that happens. So we're going to take galactose 1-phosphate, and we're going to kind of, it's going to swap out with the glucose, making glucose 1-phosphate, and the UDP and galactose are going to go together. So at the end of this reaction, we're going to have glucose 1-phosphate and UDP galactose. It's just switching it out. So, and that's with galactose 1-phosphate uridyl transferase. And that's just the uridyl whatever is with that UDP. Need to know that enzyme. Need to know that enzyme. So we've got glucose 1-phosphate now. So we're going to try to make this glucose 6-phosphate, right? Glucose 6-phosphate, is that a part of glycolysis? Yes, right? It's the second thing that we make, or really the first thing we make. And that's just going to be with phosphoglucomutase. So mutase, right, it's just kind of rearranging it, we learned. So it's going to take it from glucose 1-phosphate to glucose 6-phosphate. Really simple. So a lot of these are only going to have like 2, 3, maybe 4 enzymes in them. They're all going to do something slightly different, but really simple once you get it figured out. So galactokinase is specific. It's not just hexo, so it's going to go on that first carbon. So we're going to make galactose 1-phosphate. We don't want galactose 1-phosphate. That doesn't go into glycolysis, no? Yeah, right, it doesn't go into glycolysis? No. Well, okay, so let's get it more like glucose. So we're going to make it glucose 1-phosphate, and that's going to be with a transfer reaction with galactose 1-phosphate uridyl transferase. So we're going to take the galactose, is going to go with the UDP, glucose is going to go with the 1-phosphate, making glucose 1-phosphate, and UDP galactose. And then glucose 1 to glucose 6 with phosphoglucomutase. Then glycolysis.
What is this over here?
UDP galactose 4 epimerase. So this is a way to turn UDP galactose into UDP glucose so that we can kind of keep having that cycle going of making galactose 1-phosphate into glucose 1-phosphate. So this is just kind of its way of turning about making this again so that this other reaction by the transferase enzyme go. Question?
So is the galactose 1-phosphate and the UDP glucose, are those happening at the same time? Switching down to?
Are you saying this step right here?
Yeah.
So let's just put it this way. So we've got UDP glucose over here. This UDP glucose is going to steal the 1-phosphate, making glucose 1-phosphate. And so then UDP is like, well, that's kind of sad. Or I guess galactose is kind of sad, so it's like, I guess I'll settle for UDP. So that's why we get UDP galactose. So I guess, so it's a transfer. So glucose is taking, I guess it's swapping out with the 1-phosphate, and galactose and UDP are then going together. Does that answer your question? Okay. Any other questions? Does that make sense? Is that pretty straightforward? Okay, so here, so usually just got to know the first enzyme for sure, what it does and what it makes. So galactokinase makes galactose 1-phosphate. And I've already said the rest of it. So now we've got mannose. Mannose is a C2 epimer. Might ask it, he might not ask it. It'd just be good to know. C2 epimer of glucose. Okay, and there's a nice little picture showing you how it is, where it's different. Cute. Okay, so mannose. This one only has 2 enzymes. Really nice. I think when he asked this on our exam, he put it like in order, like what's the correct order of the enzymes. But once again, he might just ask you what they do individually. So mannose, this one uses hexokinase. There's no mannokinase, unfortunately. Man. There might be a phe-mannokinase. I don't know. Equality. So mannose goes to mannose 6-phosphate with hexokinase, right? So hexokinase, if we see hexokinase, is going to put on the 6th carbon, right? So that's why we get mannose 6-phosphate. Really simple. Then we get phosphomannose isomerase, right? Making fructose 6-phosphate. What was the enzyme that made fructose 6-phosphate in glycolysis? Close. What's enzyme number 2? So we went from, what did we use to go from glucose 6-phosphate to fructose 6-phosphate? It was phosphohexosisomerase. Yes. Yes, so phosphohexosisomerase. Now we're using phosphomannose isomerase to do the same thing, really, just turning mannose 6-phosphate into fructose 6-phosphate. Really simple. So hexokinase is usually first. Then it's going phosphomannose isomerase, making fructose 6-phosphate, which then you can have another phosphate added to it by PFK1, making fructose 1,6-bisphosphate, and what have you. Really simple. So only 2 steps. So those are pretty much the 4 main sugars besides glucose. So we've got fructose, I guess maybe 3. Fructose, galactose, and mannose. And they all just go straight into glycolysis. It's just learning the little differences, the discrepancies between the 2, between the 3 of them that make them individual for the sugar that they have, right? Because enzymes are very specific for the reactions that they have. These are very specific. So I've said it all.
Does he hear about how many ATP are used and ADP that are produced in all of these?
Not really, no.
Okay.
No, he's not going to be tracking ATPs on this. Because they're just going back into glycolysis and then following the rest of the cycle. So in theory, we already know all the ATPs being made and all the things being made. All right, so pentose phosphate pathway. So whenever we see pentose phosphate pathway, in our head, we're immediately, that should be it. So in our head, we're immediately thinking NADPH and ribose 5-phosphate. Those should just be like the first two things you immediately think of when you see PPP pathway, because he's for sure going to ask you about it. So when y'all see those things, you'll be like, oh yeah, look, they're in there. Andrew's so smart. All right, so this is an alternative pathway for metabolizing glucose. So say, glycolysis is all taken up, I guess. It's like 35 at rush hour. Like, we don't want to take that road. Let's find our back road home. Might be a little bit different, and we're not used to it, but it's still effective. We'll get home safe, hopefully. Maybe not around here, but okay. Anyway, so it's a different way to metabolize glucose. It does not, PPP does not oxidize glucose via glucose 6-phosphate to produce ATP. Instead, this pathway uses specialized products such as NADPH, which is required for numerous reductive biosynthetic pathways, and also provides pentoses for nucleic acid biosynthesis. So we had those pentose sugars. I think pentose sugars when we were making our nucleic acids earlier in like the very beginning of this try. We even want to go back to that far. Also, other monosaccharides. I just underlined a couple. So ribulose, is it a ketone aldehyde? Ribulose makes it a ketone, right? Because it's the ulose. So xylulose is a ketone because it's ulose. Erythrose, aldehyde or ketone? Aldehyde because it's just OSE. Fructose, aldehyde or ketone? Ketone. I don't even know what any of that is, so we're just not going to ask that one. All right, so important things. Uses NADPH, provides pentoses for nucleic acid biosynthesis, and also other monosaccharide sugars. So besides ATP, cells also need reducing power of NADPH for biosynthesis. So NADPH is really for biosynthesis, which is anabolism, right? Anabolism, we're adding things together. Whereas NADH over here is for oxidative metabolism or catabolism. It's a catastrophe. If you have that cat, that always messes up your stuff. So an important thing down here, this will go up a little bit further. No, just to the side. Perfect. So this is for sure going to be a test question, for sure. Knowing these different ratios. So cells and understanding why we have these ratios in our body. So cells keep a NAD plus over NADH ratio near 1,000. So this favors metabolic oxidation or catabolism. Whereas NADP over NADPH ratio is near 0.01, which favors biosynthesis. So starting at this bottom one, who can tell me why they think that this ratio is so low? Why would it make sense for us to have, I kind of have it written there, so don't look at there, don't cheat. Or maybe cheat, it's fine. Why do you think we'd have that ratio really low for NADPH when we said that NADPH is for biosynthesis? Yeah, sorry, I use pencil. I don't like, this isn't my book, I'm borrowing it, so I don't want to write in pen. So we need to have things ready to build. So NADPH is for biosynthesis, right? So if we want to build something, we're going to need that NADPH. So it would make sense that we would have, so this is NADP without the H over NADPH. So that means that we have more NADPH than we do NADP. So really it's just saying, hey, we've got all this stuff ready for when we do need to make something, it's there. We don't have to waste time trying to make it and then building it. So it's just ready to go. Need to have things ready to build. It should say, and it needs to have NADPH ready to biosynthesize materials, like cells. So on the flip side of that, we said NADH is more catabolic. So why does it make sense that this ratio of NAD plus over NADH is really high. Exactly. So if this is for the breakdown, we don't really want to have too much breaking. I'm gonna put it from a different perspective. That's how I remember it. So when we're going through glycolysis, we're making NADH, we're making energy, right? So with this ratio, NADH is lacking. So we're going to be constantly actually going through glycolysis because we're needing energy, because we're breaking those things down. We're going to be constantly needing energy. So that's why this is so low, is because it really drives forward glycolysis. It really drives forward TCA cycle. It drives forward oxidative decarboxylation to keep trying to produce these NADHs in our body. So need to know this one is really low. This one is really high. Was that clear for y'all? Does that make sense? Questions on that? Gonna keep moving on. Definitely a test question though. Sorry, my handwriting is not very visible. Okay, so oxidation of glucose by the pentose phosphate pathway generates NADPH. Gotta know it. Gotta know it. It's just one of those word association things. It's also called the hexose monophosphate shunt. So if you see it on the test, don't freak out. It's just another way of saying it. Or the phosphoglucose something, phosphogluconate pathway. Also generates ribose 5-phosphate, right? That was the other thing I said earlier on. So ribose 5-phosphate. If you see it, PPP. And if you're thinking PPP, also think just another way to oxidize glucose. That's it. Exactly. An alternative way. Probably a better way to put it, yeah. So the first part of it consists of oxidative reactions. So glucose, 6-phosphate. Is there a picture on the next page? Yeah. Let's go to this picture. Let's go to this bottom one. All right, we started reading with picture books. We're like, that's a cat. Cool. That's what we're going to do here. So we're starting with glucose is going to make glucose 6-phosphate. Okay, so glucose 6-phosphate is going to be, we have a dehydrogenase reaction, right? We see dehydrogenase. What are we thinking? Taking off hydrogens in some form or fashion, right? So in glycolysis, we made NADH, right? In TCA cycle, we made 3 NADH and the 1 FADH2 with our dehydrogenases. Now we're making NADPH. So it kind of makes sense. Dehydrogenase, the same stuff carrying over, just a different way of putting it together. So this enzyme is going to take glucose 6-phosphate and make glucose 6-phosphogluconodelta-lactone. I'm sorry about that one. I don't really know what to tell you on there. So I just think glucose 6-dehydrogenase makes the delta-lactone one. I try to just shorten it. So that's step one. So now we're going to have lactonase. So we're going to break down the lactone part of it, just making glucose 6-gluconate. So really, this lactonase is taking off this last part, making glucose 6-gluconate. And lastly, we've got glucose 6-phosphogluconate, just describing what we've got, really what we had, dehydrogenase making D-ribulose 5-phosphate. So that's pretty close to what we're trying to get, right? We want ribose 5-phosphate. So the next step would be to make ribose 5-phosphate with an isomerase, right? Because that's just the ketone. This is the aldehyde, so just use the isomerase. Switches it up. And we've got it. And that can go on to make whatever it needs to make. Biosynthesize, whatever. So I don't think he's really too concerned. I don't remember him being really concerned with a lot of these enzymes, specifically, like he was maybe with fructose and galactose and mannose. But this is definitely a pathway, and at least make it make sense to you, the general order. So glucose 6-phosphate to the big long one with lactone, to without lactone, to ribulose to ribose, the bare minimum. And that's pretty much what it's going to say all over on this left side. But that's just a lot of work to read that much. Up here, it kind of talks about how they're generated, but I don't personally find that picture as helpful. But it does. The only thing I like about this is, not the only thing, but it just tells you where it goes, where the ribose 5-phosphate goes. It's to nucleotides, coenzymes, DNA, RNA. So those are the things it's going to make. Pretty helpful. Also, I think that was supposed to say ribose 5-phosphate. Did he tell y'all to change that?
Literally, Apple's website written and watch out for spelling between ribulose and ribose.
Yeah.
And then he misspelled it.
Yeah. So I'm pretty sure that this should say ribose 5-phosphate instead of ribulose 5-phosphate. That just makes the most sense to me. Does that make the most sense to y'all too? Okay. So, a couple more pages, and then we'll take a short little break before we start chapter 13. So, this is just, let's see. This phase of the pathway generates NADPH and ribose 5-phosphate. Ribulose 5-phosphate produced in this phase is used for trans-ketolase and trans-aldolase reactions to finally form glucose 6-phosphate from pentoses. So I think that's saying if we're kind of going in the reverse direction, if we're starting with our pentoses, like what they're starting with our pentose 5, ribose 5-phosphate. So we were starting with ribulose. It can make xylulose and what have you. I don't think he really asked any questions on these. But for your own, this is the backwards reaction if you got kind of lost. Pretty sure, and that can go all the way back to glyceraldehyde 3-phosphate, what have you. Here's a different version of it. What you need to know really is on this page. What? TQ City, yeah. Rip City if you're from Portland. We got Lamarcus though, so it's okay. So anyway, so NADPH production is for biosynthesis of macromolecules. Gotta know it. So that's NADPH. Ribose 5-phosphate is for nucleic acid biosynthesis. So when we don't need ribose 5-phosphate, the pathway results in 2 NADPH per glucose 6-phosphate. So what he's saying is, like, if we don't need to make nucleic acids, you know how when we just did like NADH, like 1 mole of glucose produces, or 1 mole of NADH produces 2.5 ATP on average, and then FADH is 1.5. This is the same thing. It's just 2. Typically 2. So 1 thing of glucose 6-phosphate makes 2 NADPH. Same fashion. But when we do need it, the ribulose 5 can make that to go on and make our nucleic acids. For the time. Clinical correlate, Wernicke-Korsakoff syndrome. He might ask you about this. This is like that higher level question if you actually read to the end of the chapter or if you stopped right beforehand. You'll get this in try 3, maybe, definitely try 4 with Dr. Perryman, who's awesome. So Wernicke-Korsakoff syndrome is caused during severe deficiency of vitamin thiamine. One of the B vitamins, yes. So the active form of thiamine is thiamine pyrophosphate, TPP. It's going to be a test question in try 4 as well, or try 3. And so TPP is the coenzyme required by the trans-ketolase enzyme. So a coenzyme, right, is just the thing that helps the enzyme work together, right? So we've got cofactors, which are like metals, and we've got our coenzymes, which is like our vitamins. Yes? Y'all are telling me, like, get on with it. So in this genetic disorder, mutation of the transketolase gene causes a defective enzyme synthesis having very low binding capacity for TPP. And if it doesn't bind it, that decreases the operation of pentose phosphate pathway. So if he asks you what disease decreases the operation of this pathway, you're going to say Wernicke-Korsakoff. Or if he asks you what coenzyme, you're going to say thiamine pyrophosphate, which all we'll get into, I think maybe at the end of this book, at the end of the year when you get into B vitamins. Or is that next try? Might be next try. Definitely is next try. So, Wernicke-Korsakoff syndrome decreases it. Y'all go take 5 minutes. 2, 1.
Most of the things I say are really appropriate, so.
All right, are we good to go again? Are we good to start back up? Try to get us out of here as quick as possible. All right, so chapter 13, glycogen metabolism. So metabolism is just the summation of what? All of the things making stuff and all the things breaking stuff, yeah? So it's just a summative effects. So this chapter is going to be talking about glycogenesis, genesis in the beginning. So genesis is making, or glycogenolysis. If you see lysis, it's breaking down, right? So glycogen is what? The storage form of glucose? Yes, right? So if we've got a bunch of glucose, it goes in and makes glycogen, so we can store it. So does glycogen have branches, or is it just kind of like one big, long molecule? It's got branches, right? So the branches are kind of, the branches help it to be more soluble and to be stored better. So we've got this branched molecule. The first half of the chapter is the breakdown. The second half is making it. So when we're breaking down glycogen, we're doing this why? Because we need the glucose, right? So we don't have enough glucose, so we need to take our stuff out of storage and put it into use, right? So it's almost Christmas season, so we're about to be taking out our Christmas trees and putting them up for everything, unless you're like me and you have it up all year round, because I'm classy like that, you know? So that's what we're doing with glycogenolysis, is we're breaking down glycogen because we need glucose. So we already know what we're starting with, we already know what we're ending with, which is really nice, and it's just the few steps in between. So if it's a branched, is glucose a branched chain, or is it just the thing by itself, the molecule by itself? Does it have branches or no? Glucose. Glucose does not. Glucose is just, right, it's just one, it's like a monomer. So it's not branched. So at some point, we're going to have to take off the branches of glycogen. So there's, I guess, so we're going to have to take off the branches, and we're going to have to turn whatever we took off into glucose. So that's really what we're going to be doing here. It's important to note, glycogen synthesis occurs in all tissues. However, the liver synthesizes most of it, and then the muscle synthesizes kind of the rest of it. So really, glycogen is in our liver mainly, and in our muscles, right? It makes sense it's in our muscles, right? So if we need to run away from that bear or angry girlfriend or boyfriend, like, we should be able to do that. Anyway, and so it's definitely it's in the liver and the muscle. Muscle tissue does not supply glucose to other tissues for oxidation. So the reason I say that is because the liver does. So the liver can break down glycogen and put it into the bloodstream to be taken to wherever else it needs in the body. But if it's in the muscle, it stays in the muscle. Like I said, glycogen is highly branched, contains both linear chain with alpha-4 linkage and a branch chain with alpha-6 linkage. So y'all remember that from last test? Was there an alpha-4, alpha-6 question on there? Right, so alpha-4 just means non-branched. Alpha-6 just means branched. That's all it's talking about. So next, what do we have? So we said two things that store glycogen in the muscle. So the need for ATP is what triggers the breakdown of glycogen into glucose and to put it into glycolysis to make ATP, right? So the need for ATP is what triggers this. In the muscle, that says doesn't go to blood over there on the left, doesn't go to blood. Whereas in the liver, if we have low blood glucose, so our liver is kind of looking at our whole, the blood glucose, the glucose levels in our blood, and it's saying, hey, maybe we're too high or too low. If we're too low, we need to break down this glycogen to put it back in there. So what's going to tell us if our blood glucose is too low? What I hear over there, very faintly? Insulin is if it's too high, right? So what's the thing that if it's too low? Glucagon, right? Yeah, glucagon. I heard it. I was trying to. So glucagon, right? So if our glucagon is what's going to tell us if our blood glucose is too low, and it's going to stimulate putting more glucose into it, right? So kind of how it all ties together. So removal of glucose from glycogen. So I'm just realizing I say so a lot. I'm really sorry about that. Almost did it actually. Glucose residues are removed from the non-reducing end of a glycogen chain. It might be a test question. I appreciate it and I don't appreciate it at the same time. The glucose residues are removed from the non-reducing end as opposed to the reducing end. I don't know what that means off the top of my head, but I'm guessing that the reduced end has more hydrogens, whereas opposed to the non-reduced end doesn't, but that might be entirely wrong. Just memorize that straight up. It continues to remove glucose units until it reaches the 4th glucose unit. So we've got a big chain here. This is just very general. So we've got two chains here, and it's going to remove it until it gets to this 4th one, and then something different is going to happen. This glycogen breakdown is catalyzed by glycogen phosphorylase, but there's actually 3 enzymes here. So we've got 3 main enzymes that are responsible for glycogen breakdown: glycogen phosphorylase, glycogen debranching enzyme, and phosphoglucomutase. So, dang it, I shouldn't have said anything. Should not have said anything. Trying to improve ourselves. Glycogen phosphorylase is going to take off the individual monomers from, we've got 2 long branches. It's going to take off the individual monomers until it gets to 4. And then we're going to have glycogen debranching. It's going to take off that branch. It's going to take those 4, put it elsewhere. And phosphoglucomutase is going to just make the glucose at the very end. Very.
Sorry, go ahead.
So, dang, oh my God, I should never have said that. That's so dumb of me. Glycogen phosphorylase. It gives y'all something to laugh at. I'm okay with it. Glycogen phosphorylase makes glucose 1-phosphate. It takes off each of those monomers and makes glucose 1-phosphate. And it makes however many there are up until there's 4 residues left on that chain. And the coenzyme is pyridoxal phosphate. What this looks like is we've got a chain over here, put in glycogen phosphorylase, takes off the one on the end, makes the chain shorten by 1, and glucose 1-phosphate. And that just does that however many times is needed. Pretty simple. That's just the first one. Next, we have glycogen debranching enzyme. What do you think it's going to do? Debranch glycogen. Okay. So which one was is alpha 6 linkage the branched one, or is alpha 4 the branched one? Alpha 6. Okay. So we're going to turn an alpha 6 linkage into an alpha 4 linkage. So this has 2 different enzyme activities: the debranching and then the transferase. So it's going to remove the branches and transfer it into one big long molecule, which is what is happening here. We've got the 2 branches. Our first enzyme, glycogen phosphorylase, takes each of these off individually until we're at 4, and it does it on both. And these are glucose, or what are they? They're glucose 1-phosphate, right? Glucose 1-phosphate, so not quite glucose. Next, we're going to have the debranching part of it. So it's going to take these 3, and it's going to put it onto the bottom one, or the top one, whichever way. This one's just saying the bottom one. It's going to take these 3. It's going to put it on the main chain, because this chain is going on this side. This is just where the branch is. It's going to take the 3 off the branch, put it on the main chain. Next, we're going to cleave this bond here, and that's just going to release as free glucose. So now we've got just one chain, no branches, really simple, and that's what the debranching enzyme does. And what happens after that is, where are we at? Phosphoglucomutase, which converts glucose 1-phosphate to glucose 6-phosphate, which can then go into glycolysis, because that's why we're doing this whole thing. It's because we need glucose.
So it's converting the things that they're taking off into glucose 6-phosphate?
6-phosphate, which glucose 6-phosphate then can go back up to glucose or go down into glycolysis. Really simple. Need to know the order. Need to know what each of those does. I'm pretty sure that our first enzyme, glycogen phosphorylase, then just continues to break these off individually into glucose 1-phosphates. And then our phosphoglucomutase then converts those into the glucose 6-mutase. That's what I think. Not 100% on that. It kind of just stops right there. But that's what I think. And that's all glycogenolysis really is. Or those three enzymes making glucose 1-phosphate, then glucose 6-phosphate, then glucose. Not that bad. So this whole time we just had a lot of little reactions, smaller reactions, but not too bad. Need to know the order, because you might ask you an order question on this one. Need to know what each one of these does. Need to know that this one goes until there's 4, and that these 3 get moved over on the second half of it, and that this gets cleaved as a glucose as opposed to a glucose 1-phosphate. Where does it go? If it's in the muscle, it's going to stay in the muscle. If it's in the liver, it's going to go into the bloodstream wherever it's needed. Mm-hmm. So if we have low blood glucose, it's just going to throw it into the blood, try to get those concentration levels up.
And then those glucose 1-phosphate guys, they go and get put in through their glycolysis.
Or it could, if it's in the liver, it can go into the blood, really. So the glucose 1-phosphate can get converted into the glucose 6-phosphate, which can then be made to glucose into the blood if it's in the liver, or glycolysis if we need to break it down. Yes. Really.
And then those little yellow guys, they go back onto the blue guys through the transferase. Transferase, right?
Yes. Yes.
What are they doing now?
So this was where I'm saying I'm pretty sure that these just keep getting removed by that first enzyme, the glycogen phosphorylase. Pretty sure glycogen phosphorylase just comes in and keeps taking those off. Exactly.
The fact that somebody discovered that.
So our reaction, are there any questions on that? Any questions? Is that too difficult? If it is, that's okay. Let me know. Knowing that the debranching enzyme has the 2 functions. You need to know it has the 2 functions. The shortening of the branches, and then the actual cleavage of the branches, making it 1,6 to 1,4. Now we're just going to talk about this last enzyme for a minute. This reaction converts glucose 1-phosphate to glucose 6-phosphate. It contains a serine hydroxyl group at its active binding site. Its seryl hydroxyl group is phosphorylated in its active form. So in its active form, it's phosphorylated. Now during the catalyzed reaction of the seryl phosphate from the phosphoglucomutase is transferred. Oh my God. We're just going to skip that. It's fine. Don't worry about it. Don't worry about it. So this is just explaining kind of how all this happens. Y'all can look at that. That looks fun. Hydrolysis of glucose 6-phosphate for glucose 6-phosphatase. This is actually important. Not that the other isn't, but this is important. So glucose 6-phosphate is produced in the cytosol of the hepatocytes. Why do you think it's important that it's in the cytosol and not somewhere else? Yeah, glycolysis happens in the cytosol too, right? And also, if it needs to leave the cell, it can just go through the plasma membrane as opposed to through an organelle's cell membrane in addition to all that. So it can be transported to other tissues into the blood circulation. Glucose 6-phosphate is an ionized molecule, and thus the membrane is impermeable to this and cannot cross the plasma membrane. So it's got a charge on it. It's going to be attracted to that polar head, so it can't go through, right? Because it's going to get caught. Question?
So is this the same glucose 6-phosphate that we've made before from the phosphogluconeogenesis, and it's just like continuing on? Or
Yes. Yes. The same one, the same glucose 6-phosphate we just made. It can't go through. However, just like there was in the last chapter when we had our what was our transporter system that we did last week? Who can tell me what we did last week? Malate aspartate shuttle, and the other one.
Oh, the cytochrome, the complex 1, 2,
Oh, yeah.
3 complex.
I'm thinking more. Glycerophosphate shuttle sounds what I'm looking for. Glycerophosphate shuttle, right? So we had our shuttle systems. Here, malate, glutamate, and glycerophosphate. This is kind of similar in that it's taking it from inside the cell to outside the cell. So the membrane is impermeable to glucose 6-phosphate. It can't get out. It needs to leave. It needs to get into the blood, so it's in the liver. It needs to get out into the bloodstream to increase our concentration levels, but it can't. However, the endoplasmic reticulum membrane has a special transporter. It's so special it gets the name T1. Doesn't sound very special at all. Whatever. It has a special transporter for this, and so as a result, it can enter the lumen where G6P can be converted to glucose by the catalytic action of the membrane-bound glucose 6-phosphatate. All that's saying is T1 takes it into the endoplasmic reticulum. So go back on the putting into the bloodstream. It's going into the endoplasmic reticulum lumen. T1's taking G6P from outside, from the cytosol, into the lumen. Then glucose 6-phosphate is going to convert G6P to glucose. Then we're going to have special transporter number 2, T2, then put it out back into the cytosol where it can then leave into our bloodstream via GLUT2. So we've got 3 different kind of doorways here. We've got T1 letting G6P come into the endoplasmic reticulum, because the endoplasmic reticulum has this nice little enzyme, glucose 6-phosphatase. Phosphatase is going to break it down, and it's going to make glucose and phosphate. Then those two are both going to leave through T2, door number 2, back into the cytosol. Then we have GLUT2, which is going to take glucose into the bloodstream, increasing the concentration because glucagon told us to. If we had insulin, that's too much. So insulin's going to bring the blood into here. Maybe it's going to go into these transporters and make glucose 6-phosphate, and then that can get made into glycogen. Maybe. T3 might do that. I don't know. I just made that up, so it might do that. But it makes sense that it would do that. Does that make sense? Need to know this. Need to know, really, the enzyme, the difference between T1 and T2, and GLUT2 is probably the biggest one. Also know that it's the endoplasmic reticulum as well. Does the muscle do this? Does the muscle send stuff into the bloodstream? No, stays in there. Stays in there. So this would be happening in the liver, yeah. So this is happening in Cytosolic glucose is transported to the circulation by hepatocyte-specific glucose transporter, GLUT2. So it's only happening in the liver. Is hepato liver, or is that kidney?
Kidney is
Nephro. Y'all are so smart. So that's how we get blood. That's how we get glucose into our blood. So just y'all can go impress your friends later, or y'all's classmates who didn't come to this. So we have two ways to regulate this. Do you think Dr. Sarkar likes regulation? Have y'all learned if he likes these or not? Yes, he does. These are really simple, though. Really simple. Bless you. Inhibitors. Inhibitors of this pathway are energy, ATP, glucose 6-phosphate, and glucose. So why are we breaking down glycogen in the first place? We need glucose because glucose can then be broken down to make energy, right? So if we've got ATP, let's just keep it stored. We don't need it. Keep the, if it's not December, keep the Christmas tree in there, unless you're tacky like me. So ATP inhibits it. Glucose 6-phosphate also inhibits it, right? Because glucose 6-phosphate is the end game of what we just said. So we made our glucose 1-phosphates here, and we converted them into glucose 6-phosphate, yes, here. So if we have glucose 6-phosphate, if we have that end result, yes, it can already get into glycolysis, so we don't need to make it to put it into glycolysis. It's the same thing. If we've got ATP, we don't need to make ATP. If we've got glucose 6-phosphate, we don't need to make glucose 6-phosphate. If we've got glucose, why are we breaking down glycogen? We don't need to. We've already got glucose, so just keep it stored. So those are the things that inhibit glycogen breakdown. The things that activate it are AMP. Is that ATP? Can we break this apart to get energy? No. So we need to start making some ATP. So we need to break down glycogen to make that glucose 6-phosphate to convert into glycolysis, or to convert into glucose and make our energy. Really simple. Usually ATP inhibits most things, breakdown-wise, to get energy. We'll see maybe later it might do something different.
So if we have too much of the glucose, can we store those as glycogen in the liver?
Exactly. That's exactly what we can do. So if we have too much glucose, we're going to actually be making glycogen because there's too much of it. So we need to store it. It's like the squirrel trying to do its little thing with the acorns so it doesn't die over winter. It's going to store it. It's got a lot of them, hopefully enough. Questions on that? Important thing down, kind of hidden within here, is so the phosphorylase enzyme exists in two forms. It's got an inactive form, which is dephosphorylated, and an active phosphorylated form. Need to know the difference between those two. So, sorry. The dephosphorylated one is inactive, and the phosphorylated one is active. I think that is over here a little bit better. Phosphorylase B, less active. Phosphorylase A, active. And this is just the basic layout. Okay. This picture just kind of shows a general, or I guess a big overview picture. So glucagon or epinephrine is going to tell us that we need glucose to make energy, or we need glucose in the blood, what have you. Those two are going to be our stimuli. They're going to go through a G protein receptor, G protein membrane receptor, coupled receptor, GCPR. Let's do the acronym. So GCPR, adenyl cyclase, And really, this is just saying how much it's amplified. So 1 thing of epinephrine or glucagon makes 10,000 molecules of glucose. That's pretty cool. So that's pretty cool. I can speak for itself. That's the end of glycogenolysis. That's the end of glycogenolysis. Now we're going to go into glycogenesis. Genesis means what? Formation of, making of. Now we need to make glycogen. So if we have, why would we make glycogen? Why would we make that? Why would we make glycogen? We're storing, right? So we have too much glucose, yes? Or we don't have an energy need at the moment. We've got sufficient. Starts with the conversion of free glucose to glucose 6-phosphate catalyzed by hexokinase. Seen that before, yes? What else that we've talked about today uses hexokinase? What other things have we talked about today that uses hexokinase? Either fructose, mannose, or galactose. I heard mannose and galactose. Is that what we're thinking? I think fructose does. When it goes into the muscle. Fructose into the muscle uses hexokinase because the liver is a more specific, is a specific organ. So, God, I'm working on that. I promise you. Promise you. Hexokinase was with fructose and not galactose, because galactose is galactokinase, but mannose. Fructose and mannose are the ones that use hexokinase breaking down wise. Now we're seeing hexokinase over here in a biosynthetic pathway, just reusing things in our body so we don't have to make more, take up more space in our body if we don't need to. Just being efficient with what we've got. Need to know which ones use hexokinase, which ones don't. Need to know it's galactokinase specifically or fructokinase in the where? The liver. Fructokinase is in the liver. What was that question? You need to know which pathways use hexokinase versus like galactokinase specifically or fructokinase specifically with fructose going into the liver for breakdown. Pretty much what we talked about last chapter. Does that make sense? Okay. I feel like I have my back turned to y'all sometimes. I apologize.
Does it make you nervous?
It doesn't make me nervous. Should I be nervous? Do I need to be nervous? Now I'm watching out for y'all. I don't like it. Okay. First reaction. Hexokinase turns glucose into glucose 6-phosphate. Then glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase. Why is this just the reverse of what we just did, right? Really simple. Is that the same enzyme that we used also? Let's find out. Phosphoglucomutase. Phosphoglucomutase. Really simple. So if you see anything that goes from glucose 1-phosphate to glucose 6-phosphate or glucose 6-phosphate to glucose 1-phosphate, it's the same enzyme. So hopefully you can remember that. Now we've got glucose 1-phosphate. What are we doing next? Once the glucose, we've got glucose. So we went from glucose to glucose 6-phosphate, then glucose 6 to glucose 1. So now we've got glucose 1-phosphate. Where are we at? Now we've got 3 enzymes that convert glucose 1-phosphate into glycogen. We had 3 enzymes breaking down glycogen. We've got 3 enzymes making glycogen. We've got another one of these UDP thingamajiggers, UDP glucose pyrophosphorylase, glycogen synthase, that one's a lot nicer, glycogen branching enzyme. With our UDP, do you think that's going to be a transfer reaction again? Probably. Probably. Then we're going to make our basic form of glycogen, and then we're going to branch it and store as much as we can. UDP glucose pyrophosphorylase. Before that, what are the 3 enzymes for the breakdown of glycogen? What were the 3 enzymes that we just talked about previously? They were all on one page, nice and...
Glycogen phosphorylase, glycogen debranching enzyme, and phosphoglutamate.
Yes. Understanding the difference between the enzymes that are for the breakdown of glycogen versus the enzymes that are for the synthesis of glycogen will save y'all a lot of trouble come test day. That will take studying, but just, yeah, you might have to study just a little bit for a test. All right, so the first enzyme, UDP glucose pyrophosphorylase, catalyzes the reaction between glucose 1-phosphate and uridine triphosphate to form UDP glucose and pyrophosphate. Is there a picture of this? Is that this one on the bottom here? It is not. So there's no picture with this one. Sorry. What's happening? We've got glucose 1-phosphate plus UTP yields UDP glucose plus a couple inorganic phosphates. Not too difficult. Transfer reaction. So the hydrolysis of the cleavage of those phosphate bonds is what's providing the energy for this reaction. We're breaking bonds to release energy to use it. That's what's happening here. That's the first step. Pretty simple. Now we've got glycogen synthase. The enzyme glycogen synthase catalyzes the transfer of the glucose residue on the UDP glucose that we just made to a non-reducing end of a branched chain molecule, making alpha-1,4 glycosidic linkages, and thus the glucose chain is elongated by 1 unit. What the heck did I just say? Y'all didn't even, okay, it was above it, so y'all saw what I saw. We're taking that UDP glucose that we just made, previous step, with, what was the enzyme? UDP what? Everyone say it. Everyone get the words in your mouth. Or I guess out of your mouth. We're taking the UDP glucose that we made with that enzyme, and we're attaching it onto a chain, onto the reducing end or the non-reducing end? Onto the non-reducing end, thereby elongating the chain by +1. You're asking, where did we get this chain from? We'll eventually get there, but in this scenario, we've already got a short chain of 4 or so glucoses in a row, and we're just adding on to this chain, making a longer chain, and eventually we'll branch it with the next enzyme. So that's what we do. Glycogen synthase is just putting on, making alpha-1,4 linkages. Alpha-1,4, branched or not branched? Not branched. Not branched. Alpha-1,6, branched or not branched? Branched. All right, so do you think that glycogen branching enzyme is going to deal with alpha-1,4 or alpha-1,6? Alpha-1,6, mainly, because it's making those branches, correct? Understanding those differences can be very, very helpful in placing yourself where you're at. So that's step 2. Do that a few times. Lastly, we have glycogen branching enzyme. Alpha-1,6 linkages are formed by this enzyme, also called amyloidoglycan A14 to 16 whatever. He'll just use, I'm pretty sure he'll just use glycogen branching enzyme. Transfers 6 or 7 residue segments from the end of a 1,4 linked chain to the 6 group on a glycogen residue on the same chain or another chain to make 1,6 linked chain. What the heck does that mean? What are we looking at here? It takes, I can't figure out where I'm at right now. It takes this chain of at least how many? 6 to 7. It takes these ends, comes and attaches it over here, making the first branch. Now we've got multiple branches, our glycogen. In theory, we could then use the enzymes for the breakdown to cut those off and make what? If each one of these comes off to make what? With glucose 1-phosphate, yes? 1-phosphate until we get to 4. And then our debranching enzyme is going to put that back here and then take those off. And then we're going to turn the glucose 1-phosphate into glucose 6-phosphate, yes? Now what we're doing is we're trying to make glycogen. How we're going to do that is by taking this chain, attaching it over here, making an alpha 1,6 linkage. Really, it's just saying that it's attached to the 6 carbon of the alpha carbon, or the 6, yeah, of the alpha molecule. And that was definitely, was that a light bulb?
There was a light bulb. Yeah.
That's awesome. And we're making that glycogen now branched. Glucose residues can then be added to the new branch further by glycogen synthase, which was the second enzyme, yes? So we do the UDP. I got to learn this one. I got ARE coming out. UDP glucose pyrophosphorylase makes, it makes UDP glucose. Glycogen synthase puts that UDP glucose onto our glycogen chain, elongating it. Our glycogen branching enzyme makes our branch, and then we go back with this glycogen synthase, adding more glucose to the chains, further elongating them as needed. And that's how we make glycogen. We're doing it to make it more soluble, increasing the number of non-reducing ends. That means we can, instead of just adding glucose onto here specifically, we can add it onto here and here, making it easier to store. Increases the number of sites accessible to glycogen phosphorylase for breakdown or for synthase. If we need to store more, makes it easier. If we need to break it down more, we have different sites so we can break it down more, making glucose more accessible to us in the body. That's why the branches are so important. Questions? Are there any questions up at this point? Is this making sense? Okay, good. Glycogenin primes the initial sugar reactions in glycogen. This is what I'm talking about with the how you get the initial, those 4 segments of that glycogen chain initially. I'm not the most confident of myself understanding this point yet, so we're going to do a little bit of co-learning together right now, which is nice. I went over it beforehand, but I don't have it like straight memorized like I do other things. Just letting y'all know. I believe in like that kind of upfront stuff. I'm not just going to like bullshit y'all. So glycogen synthase, I mean, I'm not going to BS y'all, or I'm not going to pull y'all's leg. Glycogenin primes the initial sugar residues in glycogen. Need to know glycogenin. He underlined it and bolded it. And I put a TQ next to it, so you need to know it. It was an MBQ? That's what I'm talking about. Yeah, heck yeah. So glycogen synthase can't initiate a new glycogen chain de novo. Do y'all know what de novo means? from scratch, pretty much. That's what de novo means. It needs a primer. A preformed alpha-4 polyglucose chain or branch having at least 8 glucose residues. Glycogen protein serves as both the primer for new chains to be assembled and as an enzyme that catalyzes their assembly. Pretty important. So how does it work? Glycosidic bond formation between the glucose of UDP glucose. What enzyme made UDP glucose? UDP glucose pyrophosphatase? Was that what it actually is? Pyrophosphorylase. UDP glucose pyrophosphorylase is what makes UDP glucose. So it's going to make a bond between that UDP glucose and the tyrosine residue of glycogenin. Those two are going to come together. And the addition of several glucose residues, up to 8, forms the primer. So start with glycogenin, add a UDP glucose to it, makes glycogenin plus glucose, really, and then add on additional glucoses. Up to how many? 8. And that's what this picture is showing here. Here-ish. All right. This one's all color-coded, which is nice. So we start with this initial, here's our glycogenin. And we make our 8. But then from there, we can have a second tier going this way, third tier branching further and further and further and further. And that's what our glycogen is going to look like. That's how it's added in tiers. And that looks pretty organized, actually, and it is. So there's a rhyme and a reason to how it's made, because if it's made like this, really organized, it makes it really easy to take it off to make glucose and get that. It's like when you clean your room and you know where everything's at. You don't have to like look through everything. Exactly. Mom, I lost my wallet. Well, where did you have it last? I don't know. It was in my pants. Where are your pants? I don't know.
That's one, like, glucose residue.
One.
It's not glucose.
Glycogen. So we're making glycogen. That's the primer. So those 8 glucose residues really are the orange or yellow, whatever color it is.
Are there 8 on each tier?
Kind of looks like it. It kind of looks like it.
So everywhere they connect, is that the alpha 1, 6?
Pretty sure. I'm pretty sure.
Trying to make it all connect.
Good. Glycogen synthase is regulated by covalent modification. Need to know this. Phosphorylated glycogen synthase B is inactive. But this one is phosphorylated. Apo taco, whatever that means. It's on the record now. It's on the record. So when we were over here, we said that regulation of glycogen phosphorylase by covalent modification, we said that phosphorylated, what is it? Where are we at?
Right above that, does it
What?
like jump up?
This one. Oh, good for you. That's better. Phosphorylated phosphorylase A is active and dephosphorylated B is inactive. It's switched over here in regards to glycogen synthase. So glycogen synthase B, also inactive, but this one's phosphorylated, whereas glycogen synthase A is dephosphorylated. There was a question on that on our exam, and I didn't study that enough, and so I just took a shot in the dark on it. You know that B is going to be inactive on both. It's just remembering that synthase is phosphorylated, whereas phosphorylase A is phosphorylated. Or A is phosphorylated here, A is dephosphorylated here. I don't know why you put B first. That's important to get that straightened out. Okay, so I was going over this beforehand with y'all, all this stuff, and I really didn't remember any of it. So I kind of think he might have just breezed by it too. Does that sound right with y'all?
We learned more about this lunch.
Okay, so this is, if y'all just want to learn more, y'all can do it. I'm going to skip it. So, yes. On the next page or up here?
So like the insulin and the epinephrine and the glucose-6-phosphate and the glucose, the inhibitors and activators is the only part you really wanted us to know about.
Cool. So y'all saw something that regulates. Maybe put a little star next to it of what regulates. Insulin and glucagon have opposite effects. Y'all are pretty smart. I think y'all got that figured out by now. Know that. I'll write a little TQ if he said to know it. So y'all can't be like, Andrew said didn't know it. Just know this little part right here, apparently. But we're going to move on because we're going to get y'all out of here. Hormonal regulation of glycogen metabolism. Insulin stimulates glycogen synthesis, right? Because insulin brings glucose where? Into the cell. And glucagon releases it. So if our insulin is bringing it in, it's probably going to make glycogen potentially or bring it in for breakdown. Where else? Hormonal regulation in the liver, glucagon. In the muscle, epinephrine. Have y'all gotten to this page yet in his lecture? Did he start on it?
He put a star next to it, and that's really about it.
Okay. Definitely need to know these pretty well.
Pretty much all it's
He had either 2 or 3 of these questions on our exam over the next 2 pages. Just because it's very clinical, you might actually see a patient who has these things. One of these, a few of these might actually show up in your pathology classes or systems pathology class. So it'd be really nice if y'all knew them now, as opposed to me having to relearn them. When I got here, von Gerke's disease, or type 1A. I don't think he's really going to ask you the types specifically. It had more to do with the names of them. You need to know really the presentation of how they're going to present into your office. So someone with an enlarged liver with a defective glucose-6-phosphatase is going to say von Gerke's disease. Or what have you. But we can just go through them. So von Gerke's disease is you have a defective glucose-6-phosphatase. What does glucose-6-phosphatase do? Did we learn that enzyme? Did we? Let's check. Did we learn that enzyme? Glucose-6-phosphatase, is that anywhere? Y'all want to help me find it maybe if it's in here? Just so that y'all know where it could potentially be at too. Page 162. Glucose-6-phosphatase. Where? 163. Glucose-6-phosphatase. There we go. Glucose-6-phosphatase. So that turned glucose-6-phosphate into glucose and the phosphate group, correct? In where? The endoplasmic reticulum, yes? And it's going to go through the GLUT2 transporter. If we don't have glucose-6-phosphatate, we're probably going to have an accumulation of glucose-6-phosphate, yes? If we have, so these are accumulating in the liver, which could potentially lead to an enlarged liver. It is building up, not being able to get out, via glucose via the GLUT2 transporter, enlarged liver, and potentially kidney failure. Not quite sure why kidney failure, but if you see it, that's what it is. Yeah, potentially. That's von Gerk's disease. Pompei's disease is defective or deficiency of lysosomal A1-4 and alpha-1,6 glucosidase. Where do we see that? So this was page 163. Nope, this was page 163, I'm sorry. Where do we see alpha-1,4 and 1,6 glucosidase? 161, potentially. Let's go to 162. Here's alpha-1,4 glucosidase is in charge of taking that last free glucose off of that branch. So we're breaking down glycogen on this page. It's taking that last glucose off, making just the 1,4 plus the free glucose. That's what 1,4 glucosidase does, and 1,6. Oh wait, I'm sorry. The alpha-1,6 does that. The alpha-1,6 does that, I apologize. And the alpha-1,4, what does alpha-1,4 do? Anyway, alpha-1,6, alpha-1,4, enzyme degrades glycogen. Primary organ affected is skeletal muscle. Pompei's disease, skeletal muscle. That's a test question later, national board question. Can lead to muscular dystrophy or cardiomyopathy. Your heart's a muscle, yes? If you see Pompei's disease, you're thinking muscle, either skeletal or cardiac, and that is because of deficiency in alpha-1,4 and alpha-1,6. That was page 162. Forbes or Corey's disease, or dextrin, whatever. I think he had it as Corey's, if anything. Absence of debranching enzyme. If we don't have debranching enzyme, there's an accumulation of branched polysaccharides, yes. If we can't debranch it, they're just going to build up. That's going to affect the liver because why? Exactly, it's glycogen stored in our liver. If we can't break it down, it's just going to build up in the liver, which could also lead to an enlarged liver. It can affect skeletal muscle and cardiac muscle as well. So how this differs is due to the absence of the debranching enzyme. Here. Okay, Anderson's disease, or amylopectinosis, you'll for sure see this with Dr. Perryman, is the absence of the branching enzyme. So that's how it's different here. It doesn't have the branching enzyme. Is that talking about making glycogen or breaking down glycogen with branching enzyme? Making glycogen. Debranching is you're taking off branches. Glycogen is branched, trying to make glucose. Branching is you're trying to make branches, you're trying to grow a tree type deal. Trees have branches. That was where I was going with that. This enzyme causes accumulation. This enzyme causes accumulation of branched polysaccharides. How the heck does that?
Would it be
I feel like that should be non-branched. Ooh. Can y'all make sure he says that in class tomorrow if he goes over it? Can y'all ask him about that? Y'all are in charge. I don't know. Y'all. That's a question for y'all to ask him tomorrow. Y'all have class tomorrow with him?
Yeah.
Good. Ask him if that should be non-branched or not. Primary organ effect, liver. Once again, we get an enlarged liver, and we can get myoglobin in the urine. So that's different from everything else. So if you see myoglobin in the urine, you're thinking Anderson's or amylopectinosis. McArdle's disease, Also, if you see McArdle's, you're thinking muscle. So this is the absence of muscle phosphorylase. Muscle has high levels of glycogen, diminished tolerance to exercise. So I remember this with McArdle's. Ardles looks like hurdles. Hurdles are difficult, also exercise. So if you're a hurdler, you're exercising, right? Maybe, unless you're the hurdler who just kind of stood on the side and like watch everyone else do it. Maybe, no, whatever. I wasn't very good at track. Tried to do hurdles. They didn't let me do hurdles. Primary organ affected, skeletal muscle, exercise-induced cramps and pain, myoglobin in urine. So both of these have myoglobin in the urine. And that's our last one. We've got 4 or 5. We got McArdle's, Anderson's, Forbes, Pompeii's, von Gerk's. We've got 5 different ones. Understand the differences. Know the enzyme, how they could present into your office. Bless you. So key concepts. Glucose 1-phosphate is a metabolic link between glycogenesis and glycolysis. What is the link between fats, carbs, and lipids? Or I'm sorry, fats, carbs, proteins? Say it louder. Acetyl-CoA, right? That's our link between those. The link here is glucose 1-phosphate. If we have glucose 1-phosphate, we can either make or break down glycogen. Excess glucose is converted to glycogen primarily in the liver and muscle tissues when we have enough ATP. If it's in the muscle, is the glycogen going to go anywhere? Or is the glucose going to go anywhere when we break it down? No. If we're breaking down glycogen in our muscle, it's going to stay in our muscle. If we're breaking down glycogen in our liver, it could potentially go into the blood via what transporter? I'm thinking more GLUT2. I'm thinking more GLUT2 specifically. But I appreciate the effort. That was good. I'll bring, that's what I forgot. I was going to do that. It took me like 11 weeks to remember. I was going to bring little like candies for y'all and be like, here's a candy. So next week, there might be candy for people who answer questions right. If y'all remind me, remind me. It's only taken me like 4 months to forget that. Glycogen is degraded to yield glucose. Glycogen is degraded to yield glucose when tissues need ATP, mainly in muscle tissue during exercise or contraction. Also in the liver to supply the other tissues via the blood circulation. And we just said it's going to go into the blood via GLUT2. UDP glucose, last thing, the glucose unit donor in glycogen biosynthesis also donates UDP produced here as a byproduct to feed the UDP gluconorate biosynthetic pathway, which could eventually lead to form conjugated products with drugs and other stuff. He's not going to ask you about that. Gave y'all 13 minutes to spare. That's what I'm talking about. Questions, comments, concerns, compliments.
Hey, do you remember what chapter this test goes to? I know it's probably not the same for each chapter. For him, it might be.
It's pretty consistent, usually.
Right.